Hello and welcome to the webinar. Today, we will talk about rapid quantification of m6A epitranscriptomics at a single base resolution. So what is epitranscriptomics? For DNA genome, we have DNA and chromatin modifications that make up epigenome to program gene expression. Likewise, RNAs in the transcriptome are specifically modified by many chemical groups to form epitranscriptome. Epitranscriptomics is about regulatory chemical modifications on the RNAs. These modifications are not random. They are installed at the defined sites. They are evolutionarily conserved. They are inducible, reversible, and dynamically regulated. They are not about RNA sequences. They modulate genetic information flow and functions. Similarly, proteins in the proteomes are also modified with chemical groups as epiproteome. Among the RNA modifications, m6A is the most prominent epitranscriptomic modifications on mRNA. The m6A modification is usually installed in the DRACH, or more narrowly, RRACH motif sequences, while D stands for base A, G, or T, and R stands for A or G, and H stands for A, C, or T. On average, there are about 3 to 5 m6A sites per 1 kilobase length of mRNA. These modifications are enriched around the stop codon in mRNA, or in the last exon in IncRNA. m6A sites are over in the locations of mRNA where many regulatory features reside, such as splicing, microRNA binding, translation, and stability. Not all DRACH sequences are actually modified in the cells, among which only about 20% DRACH sequences are known to be modified. Therefore, additional m6A codes slightly outside the DRACH also help to determine the modification specificity. Epitranscriptomic modifications are fundamentally important in biology. For example, during development and cell differentiation from one cell type to other, there are profound changes in the transcriptomes. Epitranscriptomic modification marks rapid decay and removal of the old transcriptome for turning over to new transcriptome. In the cells, m6A modification is installed by m6A writers such as metal-3 and metal-14 methyltransferases. The m6A modified RNAs are recognized by m6A readers such as YTH family proteins DF1, 2, 3 that determine the fate of mRNAs for translation or for decay. m6A modification is reversible. m6A can be erased by erasers such as AlkBH5 and FTO in 2 steps. First, by hydroxylation into hydroxy-HM6A, and then to formyl 6A or F6A. F6A is unstable and spontaneously decarboxylated back into a modified adenosine A. So what are the molecular and biological functions of m6A modification? In the nucleus, m6A modification in the transcript is read by YTH-DC1 or serine arginine splicing factor SRSF2, which determines the splicing of the intron and alternative splicing. And 6A modification also causes RNA stem loop structure to change. This m6A switch is read by hnRNP-C and hnRNP-G readers. m6A modification is also read by YTH-DC2 readers to regulate RNA nuclear to cytoplasmic export. Newly transcribed RNAs are vulnerable to exonuclease exosome complex. m6A modified transcripts can bind to IGF-2BP and get stabilized. In female cells, m6A modified IncRNA axis can initiate X chromosome condensation and inactivation. In cytoplasm, m6A can promote translation efficiency. mRNA translation is initiated by forming a non-covalent loop among the 5' cap initiation complex and poly-A binding protein PABP. m6A reader YTHDF1 is a component of that complex. In addition to activate the translation, the poly-A tail and the m6A near stop codon can both quality control the mRNA being physically intact for full-length protein translation. The other major function of m6A is to mark the RNA for decay. Once m6A is bound by YTHDF2 reader, the mRNA poly-A tail is shortened. The mRNA is then quickly degraded in protease complex such as P-body. m6A marked degradation is an important mechanism of rapid transcriptome turnover during cell differentiation. If we look at m6A sites more closely, on average, there are about 3 m6A sites per mRNA. Most mRNAs are modified at only one single m6A site, whereas some mRNAs can have up to 20 or more m6A sites. Based on m6A site configurations along the mRNA, m6A sites can be classified into single, poly, and clustered m6A sites. A single site is an m6A site with no other m6A sites nearby. A polymethylated site has multiple individual m6A sites close to each other, at least less than 20 nucleotides apart. If a poly-A site has another m6 site within 100 nucleotides, they are called clustered sites. The single, poly, and cluster sites have functional significance. For single m6A sites before the stop codon in mRNA, it can recruit translation initiation factor EIF3 and 4TS ribosomal subunit to start protein translation, even without a M7G cap. The capless translation is a striking feature of heat shock protein mRNA during heat shock response. For a single M6A site within a codon, it can slow down tRNA loading and translation elongation. This can have consequence of protein product folding. For a single M6A site near stop codon, YTHDF2 reader can lead to mRNA degradation and decay. Here is an illustration of how single M6A in the codon can alter protein translation function. In eukaryotic cells, protein translation and protein folding are coupled. An M6A modification in the codon can slow down and store protein translation. During this brief pause in time, nascent peptide chains can be chaperoned and folded differently, resulting in different functional isoforms or subcellular localization of protein products from the same gene. Compared with single M6A site, poly-M6A site has much higher affinity to its reader such as YTHDF due to its multiple binding valency. In the synthetic RNA without an M6A, the RNA is completely in the homogeneous solution. With multiple copies of M6As, the RNA in the presence of YTHDF protein are partitioned into liquid droplets under the phase contrast microscope. In the cells, this RNA phase separation is a required part of the mechanism for RNA partitioning into subcellular components, such as stress granules under cellular stress or P-bodies for RNA decay. The location of M6A in different parts of the mRNA regions also serve different functions. M6A in 5' UTR can initiate capless translation during heat shock. M6A encoding sequence CDS or 3' UTR can be either recognized by YTHDF2 for mRNA decay or insulin-like growth factor 2 mRNA binding protein IGF2BP for mRNA stabilization. m6A in 3' UTR can also bind YTHDF1 and metal-3 to form an initiation loop to the 5' mRNA to constitutively activate translation in cancer cells. But the activation needs additional stimulus such as axonal regeneration and neuronal activities in neuronal cells. As in mRNA, m6A is the most abundant modification in non-coding RNAs or lincRNAs. LincRNAs do not have protein coding sequence, so m6A distribution does not concentrate near the stop codon that does not exist to begin with. As in mRNAs, lincRNAs can have local stem loop structure called m6A stretch, where m6A modification can weaken the base pairing in the stem and expose buried poly-U5 or 5-uridines for HNRMPC binding. The binding affinity with m6A is 8 times stronger. It is interesting to point out that HNRMP readers do not directly bind to m6A. Rather, the reader binds the adjacent poly-U5. In IncRNA MALAT1, m6A switch regulates its RNA splicing in abundance, which leads to lung cancer progression and metastases. m6A switches are not isolated cases. They are widely found in about 39,000 RNAs. In IncRNAs, m6A modification also plays cellular functions. In IncRNA RNase link 1281, the single m6A site is essential for strong microRNA LET7 binding, which acts as a microRNA sponge to competitively prevent ln26 mRNA from being silenced. ln28 protein is effective for cell differentiation. Without m6A modification, LET7 is released from the lincRNA and the ln28 mRNA is silenced. With that, the differentiation of cell is restricted. So this is an example of m6A regulating IncRNA as a competing endogenous RNA or cRNA. Just as m6A can regulate mRNA decay, m6A can regulate lincRNA stability. In dendritic cells, CCR7 chemokine receptor activates m6A demethylation in lincRNA DPF3 and prevents lincRNA DPF3 from degradation. lincRNA DPF3 inhibits hypoxia-inducible factor HIF1 alpha glycolysis, dendritic cell migration to lymph nodes, and termination of dendritic cell-based immune response. The molecular functions of m6A are associated with pathophysiology of human cancers and diseases. For example, m6A writers metal-3, metal-14, erase FTO, and read YTH can have MYC, BCL, NANOG, AKT, SOX2, and many other oncogenes and anti-oncogenes mRNAs as the target RNAs. m6A modifications are causal factors in acute myeloid leukemia AML, bladder cancer, breast cancer, colorectal cancer, glioblastoma, hepatocellular carcinoma, and lung cancer. m6A modifications are also associated with many other diseases. For example, m6A eraser FATO actually got its name from fat mass and obesity-associated protein, with SRSF2, RANKS1T1, and many other RNA as the targets. They are involved in epitranscriptomic alteration in adipogenesis or food intake that leads to obesity. Likewise, m6A writers and erasers are associated with type 2 diabetes, growth development delay, neuronal disorder, neurogenesis, and impaired fertility. Additionally, m6As are involved in regulation of immunity and infection. in particular hepatitis C, dengue, Zika, yellow fever, West Nile disease viral RNAs are modified by m6A and read by YTHDF2 proteins to affect viral RNA partitioning into liquid droplets and viral assembly in the cells. In this particular example, m6A controlling anti-tumor immunity published in Nature a few months ago, m6A modification and its YTHDF1 reader control the target cathepsin mRNA translation. Higher m6A level increases the translation of cathepsin. The increased cathepsin protease degrades the captured tumor antigen in the dendritic cells, preventing antigen presentation to CD8+ cytotoxic T cells for tumor killing. Tumors like these are called cold tumor, which are refractory to immunotherapy. If you lock out m6A reader YTHDF1, less cathepsin is produced and more tumor antigen is preserved for the antigen presentation to CD8+ cytotoxic T cells. The tumors become hot and are primed for immunotherapy. Now we have come to appreciate the exciting epitranscriptomics in biology and disease. So how do we profile m6A epitranscriptomics? Ideally, the profiling shall determine which individual RNAs are m6A modified, m6A modification sites in RNAs, m6A sites in 5' UTR, CDAs, 3' UTR of the RNA relating to molecular functions, the percentage of modified and unmodified RNAs or modification stoichiometry for each site, the abundant levels of m6A modification for each site, the differential m6A modification stoichiometry for each site, and the differential m6A modification abundance levels for each site. For each transcript, it is a pool of unmodified and modified RNA molecules. For each transcript, there are two key measurements of m6A modification: the abundance of m6A modified RNA and the percentage of modified RNA molecules in that transcript. The abundance and the percentage of modification numbers are both important in epitranscriptomic profiling. For each mRNA, the m6A and its readers confer different metabolic fates and functional roles of the modified and unmodified RNAs of the transcript. For example, the unmodified RNAs can be destined for protein translation, whereas modified RNAs can be destined for RNA degradation. The effect on RNA modification depends on both the molecular consequences and the percentage of modification. For example, if the molecular consequence of the modification is splicing, even a small percentage of modified RNA can have a significant biological effect, as the alternatively spliced isoforms can emerge and yield different function. On the other hand, if the molecular consequence of m6A modification is RNA decay, a small percentage of modification is not going to have a big enough impact on the overall RNA level. In this case, a larger percentage of modification is needed to cause an appreciable RNA level change. Therefore, the m6A effect is contributed by both the molecular mechanism and the percentage of modification. Although profiling m6A by mirror sequencing can measure the abundance of m6A RNA, profiling the percentage of modification has been a big challenge. The lack of modification stoichiometry has been scientists' top concern in the field, as in the commentaries in Nature Review Molecular Cell Biology. Reads, current epitranscriptomic studies deal mostly with identifying which sites are modified and not the fractions of RNAs in which each site is modified. High-throughput methods that can determine modification stoichiometry are needed to address these aspects of epitranscriptome. And in science, the potential effect on mRNA modification depends on both the molecular consequences and the percentage of transcripts that are modified. A limitation of current m6A profiling methods is the lack of quantitative information about the extent of modification. High-throughput methods to quantify site-specific m6A will considerably advance the field. Methylated mRNA immunoprecipitation sequencing or mirror sequencing has been used for m6A profiling. Although current mirror sequencing technology can map m6A sites in RNAs and measure the abundance as the peak heights, it has many limitations. It cannot profile the modification stoichiometry or the percentage of modification. It is also difficult to profile m6A modification to specific transcript isoforms due to the generally low sensitivity of transcript assembly by RNA sequencing. Furthermore, mirror sequencing requires massive amounts of RNAs of greater than 100 micrograms to do the experiment, which is not practically available under many circumstances. A much better solution to this limitation is Arraystar single nucleotide m6A microarray. The method is based on unique enzymatic activity of MazF that cuts unmodified ACI sequences while leaving m6A modified ACA alone. The cutting site is directly 5' of the unmodified m6A site. In a typical m6A single nucleotide microarray workflow, the RNA sample is fragmented and equally divided into input and MazF fractions. The MazF fraction is digested with a MazF enzyme, where the unmodified sites are destroyed and the m6A modified sites are preserved. This fraction is labeled with red-colored silane 5 dye. The IncRNA is not treated with MazF. It includes both modified and unmodified sites. This fraction is labeled with green-colored silane 3 dye. Both MazF and IncRNAs are spiked in with internal reference controls that combine together for microarray hybridization to interrogate each quantifiable m6A site. With the spiking calibration, the modified and unmodified amounts for each site are accurately quantified, and the percentage of modification is calculated. The percentage of modification is calculated by this simple formula: the modified amount divided by all RNA amount obtained from the intensities in the two color channels that are corrected by the spiking references. You do not need to perform these calculations by yourselves. They are computed internally in the data analysis pipeline. The m6A sites on the microarrays are collected by compiling all the MazF cleavable RRACA motif sequences in the entire transcriptome of RNAs and the lincRNAs. These sequences are grouped into single ACA site, where there is only one site by itself within 40 nucleotide region, multi-ACA site, where multiple sites are within 20 nucleotides within each other, and clustered ACI site, where multiple sites has another site nearby, but all within a region of 500 nucleotides. The computationally collected sequences are further qualified with prior mirror sequencing and microlith data as the experimental evidence for the site's confidence. Arraystar single nucleotide m6A microarray covers human, mouse, and rat species. Close to 15,000 m6A RRACA sites are collected. All of them are qualified either with microlith or mirror sequencing experimental data. The majority of the sites, about 2/3 of which are single sites, with about 1/4 being multi-m6A sites. The spiking, positive, and negative control probe sets are designed to ensure the accurate quantification and data quality. The m6A site conservation among the human, mouse, and rat species are flagged, as explained in the next slide. Certain m6A sites in mRNAs are conserved. For example, The m6A sites in salute carrier SLC mRNA is conserved between human and mouse to the same location near stop codon. The conserved m6A sites are more likely to have conserved biological functions. You may prioritize these conserved m6A sites for follow-up studies. The m6A microarray data are analyzed and richly annotated. They include modification stoichiometry, m6A location at a single base resolution, single, multiple, classed m6A sites, mRNAs and IncRNA coverages, 5' CDS, 3' UTR in the transcript region, m6A sites conserved across the species, DRACA motif sequence. All these are essential for understanding m6A dynamics and function. These are not available by any other techniques, such as mirror sequencing. Here, the two key measurements of m6A modification stoichiometry, such as 24% or 51% modification, and the m6A abundance for each site are calculated. The differential modification analysis is performed to compare groups or conditions for both modification stoichiometry and abundance. The differentially modified RNAs are subjected to gene ontological functional analysis and biological pathway analysis, which help to gain functional insight into m6A impacts on the biology and disease. The m6A profiles are hierarchically clustered and presented in the heat maps. The m6A microarray project is typically set up to compare two conditions, such as case versus control, diseases versus healthy, before versus after treatment. At least 3 biological replicated samples are groups for each condition. Armed with the m6A microarray data, the differentially modified m6A sites, the abundance stoichiometries are identified for further validation by MazF qPCR. These m6A sites and RNAs can be further studied for their biological functions and mechanisms of action. For example, RNA stability, translation efficiency, RNA splicing, and m6A site mutational analysis. You can also use gain of function or loss of function of the m6A writers, erasers, and readers to study the effects on these sites. Or you can use CRISPR-tethered writers or eraser to target particular m6A sites. As m6A microarray requires very small amounts of total RNAs to work with, at about 2 to 3 micrograms, you can expect m6A profiling for virtually any cell types, tissues, or clinical samples, which is not otherwise possible for mirror sequencing, requiring enormous amounts of RNAs at about 100 micrograms. Now, the m6A Epitranscriptomics research opportunity is now wide open. Here is a recap of why choose m6A microarray over mirror sequencing. Under many circumstances, mirror sequencing simply cannot be done when the available RNA sample amount cannot meet the high demands at greater than 100 micrograms, whereas m6A microarray only needs a few micrograms of RNA. Second, m6A microarrays can provide the key information about the m6A site at a single base resolution and m6A stoichiometry, whereas mirror sequencing cannot. Third, probe-based m6A microarray is highly sensitive even for RNAs expressed at low levels, for example, mostly IncRNAs, whereas mirror sequencing can profile abundant RNAs and misses out RNAs expressed low levels. Fourth, m6A sites are precisely defined at a single nucleotide localization, so qPCR validation is simplified to cover that location, whereas mirror sequencing can only locate the site in 100 to 200 nucleotide cut range, whereas many potential sites are possible, so multiple qPCR are required to confirm which ones in that regions are modified. To summarize, m6A microarrays has many unmatched capabilities and benefits. It is an independent methodology for detecting m6A, allowing for the first time systematic m6A profiling not relying on anti-m6A antibody. It pinpoints m6A sites at a single nucleotide resolution. It quantifies m6A abundance and the stoichiometry for each site. It covers both mRNAs and IncRNA. There is no need for poly-A selections or ribosomal RNA removal of the sample. It requires as little as 2 micrograms of total RNA, so m6A profiling can now be performed on samples with a very limited supply. For example, precious pathological specimens, particular histological sites, sorted cells at low yield, or small animal models. Everything above on one array per sample. I think I will stop here. Thank you and have a great day.